Hello everyone. Now we will be talking about the longitudinal section of the testis. So in the previous diagram we have already seen the testis as just the outer external external diagram. Here we have made a cross section of the entire figure. Now the testis is actually in lengthwise it is around 4 to 5 centimeters, whereas it is having a width of around 2 to 3 centimeters. Right. Now if we start with the structure, you will see that there is a scrotal skin. The scrotal skin is actually the structure of the scrotum. The scrotum, which is the loose pigmented pouch of skin, right, and this arises from the lower abdominal wall. Now if we just understand this portion, you will be able to get this information that the scrotum is like in this shape, where there is an internal division which is known as septum sertoli. Right, this divides the scrotal sac into two sacs, right, and in each sac the testis lies individually, right. So septum sertoli divides the sac, and one sac, one testis, like that. And externally also there is an indication which is termed as raphe, raphe. Now this scrotum sac, this possesses smooth involuntary muscle which is known as dartos muscle, dartos, dartos muscle. And in this figure you will be able to see that I have written the name of the structure of gubernaculum. Gubernaculum is nothing but the padded region, the padded structure which holds, or where this testis is resting on this scrotal sac, right. It is seen that in fetus the testis develops in abdomen, and then finally it descends in the scrotum around 7 months of the gestation period, right. And it passes through a canal-like structure which is known as inguinal canal, right. And if the testis descends, if it fails to descend, right, like I said that in fetal condition the testis passes from the abdomen region to the scrotum, right. But if it fails to reach into the scrotal sac, such condition is known as cryptorchidism. Cryptorchidism is that condition where it fails, the testis fails to descend in the scrotal sac, and that condition leads to sterility, right. Now one more important point is that the testis descends down in scrotum sac to lower the temperature to 2 to 2.5 degrees centigrade. This entire testis along with the scrotal sac, it hangs down so that the temperature of the testis goes down by 2 to 2.4 degrees centigrade. This is important to maintain spermatogenic tissues as well as the process of spermatogenesis, right. In some breeding mammals it is seen that this event is seasonal. The testis descends in the scrotum during the breeding season but remains within the abdomen in other season, right. Such examples are otter, then bat. But peculiarly in some other organism the testis is seen to remain within the abdomen, Such example of organisms are elephant, then whale, and in them the scrotal sac is absent. Right. Next, we move on to the particular cross section of the testis. Here you can see I have drawn the figure. Right. It is actually oval in shape and pinkish in color. Right. And it is seen that it is suspended by spermatic cord, the sperm duct. Right. If you see prominently, I have mentioned the testis is actually having three covering: tunica vaginalis, tunica albuginea, and tunica vasculosa. Tunica vaginalis, tunica albuginea, and tunica vasculosa. Now, there are number of septors which are present, and these septors are actually helping the formation of lobule like structure. You can see I have mentioned it here as testicular lobules. Due to the formation of the septa, there are around 200 to 300 conical testicular lobule. You can see the red marking which I have shown here as conical shaped structure. These are the lobule, and each lobule is filled with connective tissue with around 1 to 4 convoluted seminiferous tubules. Right. When we go to the transverse section, we'll also talk about Leydig cells, Sertoli cells, but right now we will focus on to the portions of the other structures. Right. In here, you will see that the seminiferous tubule, as it is convoluted here, they become straight toward this particular region, toward the end of the lobulated structure. Here it becomes straight, and this particular portion is termed as tubuli recti. Tubuli recti, tubuli recti. Now, tubuli recti enters into a network of fine channels which you can see I have named it as rete testis, RETESTIS, rete testis. You can see the structure of the network forming here. So tubuli recti actually forming a network of fine channels which is termed as rete testis. Right. Now you can see gradually the rete testis, it is getting connected with the epididymis. Epididymis is divided into a head region, a body region, and a tail region. Now they are named also accordingly. So the head region is named as caput epididymis. The body region is termed as corpus epididymis, and the tail region is termed as cauda epididymis. Right. Now let us come back. Rete testis is the portion which gets connected to the caput epididymis, right, by around 12 to 20 tubules, and they are the vasa efferentia. Here there are ciliated cells which are present that helps in conducting the sperms. Right. Now coming to the epididymis portion, these are, you can see, highly convoluted tubule like structure, coiled structure, which are narrow, and there, as I have discussed, that there's a head region, caput region, there is a corpus region, and there is a cauda region of the entire epididymis structure. This epididymis helps in nourishment and maturation of spermatozoa, and here sperms can be stored for like few hours to few days, in fact. And if the sperm is not ejaculated, then it can be also reabsorbed. Now, testis along with epididymis, is termed as testicle, 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 right? Now, if we come down to the portion of the semen formation, let us just come back to the structure of the transverse section, then we'll talk about the semen formation. Here you can see that I have drawn the figure of a transverse section of testis. So if we make a transverse section of testis, we will find there are spermatogonium cells which are present in a layer, right, of 4 to 8 layers of spermatogonic cells are present. This is the cross section of a seminiferous tubule. Right, in the previous diagram we have seen that the lobule comprising of seminiferous tubule. Here it is one of the cross section of seminiferous tubule. This is a cross section, part of cross section of seminiferous tubule. This is also one, here is one. So they are present. As they are present, these are actually layers of spermatogonic cells. It can be around 4 to 8 layers, and here it is, here you can see I have drawn very small pyramidal cells with apical ends towards the lumen part, right? And it is seen that these particular cells are termed as Sertoli cells. Sertoli cell is given by the name. The Sertoli name is actually because of the discoverer, which is Enrico Sertoli. And these cells are actually pyramidal, like I said, and their terminal end develop a kind of processes which helps in holding the growing spermatids. And it is also seen that these cells help in secreting spermatogenic substances, right? And that is why Sertoli cells are also termed as the nurse cells, and it helps also in differentiating spermatozoas. Sertoli cell is also found to secrete inhibin, which controls the follicular stimulating hormone secretion. Right. Now, in this particular structure of testis, you will see that I have drawn some circular structures, right, elongated structures. These are actually Leydig cells, or which we also term it as interstitial cells. Leydig cell, actually because of the discoverer again, Leydig, who has named it as Leydig cells. And here it is present in between seminiferous tubules, right? And these Leydig cells or interstitial cells, they help in secreting testosterons, right? And with this we come to the end of the portion of the testicular structure, and also with the end of the cross sections of the testis. Now let us just conclude the entire male reproductive system with the semen. The semen is actually milky. It is viscous and alkaline because of the secretions of all the accessory gland. This semen becomes alkaline in nature, and here the semen is seen to be having a pH of around 7.3 to 7.5, right? And it is seen that around 2.5 to 4 ml is actually secreted at a time per ejaculation, and around approximately 400 millions of sperms are present within that particular moment. right, in that 2.5 to 4 ml of ejaculation of semen, right. And here it is that the fructose which is present that helps in nourishing sperms. There is a presence of calcium and bicarbonate which helps in neutralizing acidity in female reproductive tract. Why? Because this actually neutralizes. Semen is alkaline and female reproductive tract is acidic, so these components it actually neutralize so that the sperms can actively do the process, right. And there is also prostaglandins prostaglandins which actually helps in stimulating the movement in female reproductive tract. With this we come